Hello friends, welcome back to our channel NEET Biology Expert. I am Dr. Parveen. In this lecture series, we are studying Class 12 Biology, Molecular Basis of Inheritance. In this particular session, we are going to discuss about the structure and functions of messenger RNA. Alright, so first, in the year 1969, two scientists called Francis Jacob and Jacques Monod. In short, we call them Jacob and Monod. So these two scientists, they proposed this idea of mRNA, which actually helps in transferring the genetic material from the nucleus of the cell, that is from the DNA, to the ribosome in the cytoplasm where actually protein synthesis occurs. Okay. So what actually they proposed? Look here. So this is the cell, this is the nucleus where the genetic material DNA is present. So here this structure is the ribosome where protein synthesis occurs. Okay. So we know that DNA gives the information to ribosome in order to produce what type of protein is needed for the cell. So these two scientists, they had this idea that how the genetic information from the DNA, which is actually present inside the nucleus, is carried and transferred to the ribosome, which is present outside the nucleus in the cytoplasm. So how this information is transferred out of the nucleus? So when they thought about this thing, they got this idea of mRNA, messenger RNA could be the reason, and they proposed this idea, okay, in the year 1961. So Jacob and Monod. Right. So first let us see what is a messenger RNA. As the name indicates, it carries messages from the DNA present inside the nucleus to the ribosome or the site of the protein synthesis. Okay. So the messenger RNA or mRNA transfers the genetic information from the DNA in the nucleus to the cytoplasm where the proteins are synthesized, that is in the ribosome. So when we look at the structure of the messenger RNA, this is the structure of the messenger RNA. This is a long linear molecule. Okay, so this is long RNA. So we have seen 7 types of RNA, right? So out of all this RNA, which is the longest molecule? The answer is messenger RNA. mRNA is the longest RNA. So which contains around 75 to 6,000 bases, right? It constitutes about 2 to 5% of the total RNA. So out of all the types of RNA, mRNA is constituting 2 to 5% in the cell, right? So this is a linear. What do you mean by linear? Linear means like the structure. So this is a linear single stranded molecule and never coiled. So what is never coiled? So we have seen previously. That means at some places whenever is needed, the RNA coils like this and form a double stranded like structure, isn't it? So this character is not applicable for mRNA. mRNA is a linear single strand molecule. It never gets coiled. Now, what is the major function of the mRNA? It carries the messages from the DNA into the ribosomes, the site of protein synthesis, right? So it carries a messages, that is the coding message. Coding message means it carries the genetic information which protein has to be produced. Okay. So this property or this function cannot be done by other types of RNA. Okay. So this is the only sole function of messenger RNA. So that's why this messenger RNA is also called as informational RNA. It carries the genetic information from the DNA. Also, this is called as genetic RNA, right? Also, this RNA is short-lived. Short-lived means the lifespan of this RNA is very small. Once it finishes its function, once it brings the genetic code and passes the information to ribosome, then it gets degraded. Once the protein is synthesized, that's all. Its function is finished. It gets degraded. Okay, it won't work anymore. So this is a very short-lived RNA. Now, let us see how this messenger RNA is produced or formed. So, where this messenger RNA is produced in the cell. So, actually, the basic concept of the messenger RNA is to carry the genetic information from the DNA to the protein synthesis site, right? So, this messenger RNA is produced within the nucleus by the DNA. Okay. So, see here. the double stranded DNA. In this picture, this blue color thing is the double stranded DNA. Okay. So here, a particular segment of this double strand DNA, it wants to encode some information to produce a protein. So what will happen? First, this double strand DNA, it unwinds and forms single strand. So this is one strand, this is another strand. Okay. So one strand acts as a template. So using this template strand as a model, now the complementary RNA is going to be produced. So this function will be carried out by an enzyme. That means it is catalyzed by an enzyme called RNA polymerase 2. Okay. So RNA polymerase 2 is the enzyme. So this orange color thing is the enzyme. Understand? So now see here in this template strand, here the nucleotide is thymine. So complementary of the thymine is adenine. Adenine will be added by the enzyme. Then the next nucleotide is adenine. So complementary to adenine, now this is RNA. So instead of thymine, here uracil will be added. So adenine pairs with uracil because this is RNA. Then guanine. So complementary to guanine, cytosine. Similarly here. Then complementary to thymine, adenine. So like this, by reading this template DNA strand, this RNA polymerase 2 produces a short segment of RNA in the 5' to 3' direction. So this RNA is now called as mRNA. This is the mRNA. So this process is what basically we call as transcription. What is transcription? Transcription is the process by which the information in the strand of DNA, in this strand of DNA, is copied into a new molecule. This is copied into a new molecule, that is the messenger RNA. Whatever information present in this strand is as such copied into a messenger. That messenger is what we call as mRNA. So this process is called as transcription. So you must basically understand this mRNA is synthesized inside the nucleus using the nucleotide sequences of DNA as the template. So now inside the nucleus this thing happens. Okay, so inside the nucleus this thing happens. This is what we call as transcription. So up to here is the cell. Okay, so now this is the cytoplasm, right? So whatever the mRNA produced here, this will travel and comes out of the nucleus and passes its information into the site of protein synthesis, that is the ribosome. Understand? So this is what basically occurring in the synthesis of mRNA. Right. So let us see how exactly this mRNA is produced. So let us zoom into the nucleus and study further. Okay. So inside the nucleus, now we are talking about only inside the nucleus. So this is the nucleus where the mRNA is produced. Okay. So this is the double-stranded DNA. So basically, when we see this double-stranded DNA inside the cell, that is the genomic DNA, this DNA is what we call as heteronuclear DNA. Heteronuclear DNA. So what do you mean by heteronuclear DNA? The DNA contains both coding sequences and non-coding sequences. Okay, introns are the non-coding regions of a gene. Introns are the non-coding regions. Okay. Exons are the coding regions. Coding regions. So what do you mean by coding and non-coding? Coding means it gives a particular information. We will study about the triplet codons in our next lessons. Okay. So coding means a triplet codon A, U, G, U, G, A. So this is one coding. This codes for a particular amino acids. This code for a particular amino acids. So they join together and form a protein. So why this intron is present? What is the function of this intron? Though introns are non-coding sequences, they are important for gene expression and regulation. During this transcription process, okay, the genetic information, whatever the genetic information coded by this double-stranded DNA, all those informations will be as such copied into RNA. Understand? So this process is called transcription. So that means here both the introns and exons, both the genetic informations are copied as such. Understand? So now this RNA is what we call as mRNA. That means this is a precursor mRNA. This is not a complete mRNA, or this is not a functional mRNA. Okay, this is called precursor mRNA or immature mRNA. Immature mRNA. Why do you call like this? Because this mRNA contains the required coding sequences, exons, and also the non-required intron sequences. Okay, so if you look at this picture here, see here in the DNA, these blue color things, these blue color sequences are the essential exons, whereas this green color thing are the non-coding sequences, introns. Okay, so when this genetic information is transcribed into mRNA, okay, so this blue color DNA information is transcribed or copied into mRNA into this red color thing. Okay, see, this is exon, 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 right? Similarly, in between you could see the green color thing. These are the introns. That means non-coding region. All this non-coding region, they are also copied to the mRNA. Understood? So in the transcription, both coding sequences and non-coding sequences were copied. Now, what is the next step? All these non-coding sequences has to be removed. So in order to remove that, endonucleases enzymes come and functions here. Okay, so the endonucleases cuts the bond between the introns, so the enzyme endonuclease come and act here, and they cut out all this unwanted introns. Okay, so what will be remaining all this red color exons, but there will be a space here because these introns were removed, so there will be a space here, right? So those spaces has to be ligated, joined. So for that, and another enzyme called ligase will come and act. So ligase joins all these exons together, so as a result here we get an messenger RNA. Understand? So two enzymes comes and act here. Endonuclease comes and cut all the unnecessary introns part. So that process is called splicing. Splicing means cutting. RNA splicing, RNA cutting. After that, there will be gaps in between, right? So those gaps will be filled by ligase enzyme. So finally, here we will have the mRNA. So this mRNA here is actually not the complete one. This is an unstable molecule. Unstable molecule. It has to be stabilized. Okay. So let us see how this is done. So here, so look at this picture, the same concept here. Here, the pre-mRNA is formed. Okay, so here first let me write, this is a double stranded DNA. This is transcribed into pre-mRNA. So pre-mRNA will have intron, exon, everything, right? So then what will happen? Endonuclease enzyme will come and cut, and it does the splicing. So all the introns will be removed. So only exons will be there. Then the ligase enzyme will come and join all the exons together. Okay. So this structure is now the mRNA, but this is unstable. Unstable. It has to be stabilized. So how the stabilization is done? That is at the 5' region and the 3' region. This is the 5' end of this nucleic acid. This is the 3' end. Okay. So at the 5' end, there will be a cap added. At the 3' end, a poly A tail will be added. So this is stabilized. That means the mRNA is stabilized by adding a cap at the 5' end and then a poly A tail at the 3' end. So now this is a stable mRNA molecule, and this mRNA molecule will now come out of the nucleus and it does its function in the cytoplasm. Okay, so this is how mRNA synthesis will occur. Now, if you see here, see here from the double stranded DNA, mRNA is produced. This process is called transcription. So this mRNA is not a complete mRNA. This is a pre-mRNA or a precursor mRNA. From this precursor mRNA, after splicing, ligase, action, everything is done. Capping, poly A tail, everything is finished. Then only it will become a mature mRNA. So this event is called as post-transcription event. Post-transcription event. Okay, understood? Understood? So after this post-transcription event only, the mRNA gets matured and it will come out of the cell. Got it? Right. Now, let us see the structure of a mature mRNA. So we have seen the cap, poly A tail, all these things. No? So what is it exactly? Let us see that. So this is the structure of a complete mRNA which comes out of the nucleus. Okay. So this mRNA contains certain things. So let us see this one by one. So first, this is the mRNA, coding sequence of the mRNA. That means this is the region which contains the genetic information copied from DNA. Okay. But in order to preserve or protect this information, it has to be protected at both the ends. That means at the 5' end, it has some protection. At the 3' end, it has some protection. So what is that? Okay. So at the 5' end of the mature mRNA, a cap is present. What is this cap? See, when we go out in a hot summer, in a sunny day, we used to wear cap, right? So what is the purpose of the cap? Is to protect our head, right? Directly from the sun rays. So similarly, in order to protect this mRNA against degeneration. Degeneration means destroying or getting spoiled. Okay. So the 5' of the mRNA is protected by a cap-like structure. So what is this cap-like structure exactly? This is 7-methyl guanosine. This molecule is present like a cap at the 5' end of the mRNA. Okay. So this structure is the 7-methyl guanosine cap. Understand? So its major function, this cap protects the 5' end of the mature mRNA against degradation or degeneration. It protects the RNA from the degeneration. And another function is it directs the binding of the ribosome during protein synthesis. So the mRNA has to come and bind to the ribosomes, right? So it directs how to go and bind to the ribosome. So the 5' cap will enter first. So like this, this is the another function, right? At the 3' end here, at the 3' end of this mRNA, it contains poly A tail. A stands for adenine. Okay, so it contains adenine bases only. Only adenine bases will be there for about 150 to 250. A like this. Okay, so what is the function of this poly A tail? Again, its function is to protect the 3' end of the mRNA from degeneration. It protects this end from getting degenerated. Okay, and second thing is it helps in the transportation of the mRNA residing within the nucleus to the cytoplasm. So this 3' end only helps the mRNA to come out of the nucleus and it helps to go into the cytoplasm. Understand? So this is the function of the 5' and 3' end of the mature mRNA. Now, the next thing is, next to the 5' and the 3' cap and the poly A tail, what is present? Here at the 5' region, there is a untranslated region which we call as UTR. Again, here at the 3' end, there is an untranslated region. What do you mean by untranslated region? This is also some gene sequence, but this genetic sequences will not be translated. That means it will not be converted into protein. Okay, it will not code for any amino acids like this. Okay, so it will not be translated. It will not be converted into a protein. So this is called untranslated region. But why this is present? This gives some stability to the mRNA molecule and it controls the ribosome's ability to bind with the mRNA. So when the mRNA goes to inside the ribosome, this region helps the mRNA to bind with the ribosome properly. Okay, this is the function of the untranslated regions. Now, next to the untranslated region, okay, inside, if you see, at the 5' end, there is a start codon. At the 3' end, there is a stop codon. So what is the start and stop codon? See, this center part is the coding region. Coding region means this region only contains the genetic information from the DNA which has to be translated, what protein has to be synthesized, right? So, how the ribosome knows when to start this protein synthesis and when to stop the protein synthesis. Okay, so in order to code that, there is a specific codon that is called as start codon. The start codon is always AUG. AUG. This is a triplet codon. Always the genetic codes will be triplet. Three nucleotides together. Okay. So AUG codes for methionine. Methionine is the start codon. Okay. So when AUG is present, then methionine molecule, methionine amino acid will be added. So when methionine is there, then the protein synthesis will be initiated. The amino acids will be keep on added. Okay, so whatever is coded in this coding region, it will be added. So when the stop codon comes, the translation or the protein synthesis stops. Okay, so what are these stop codons? Three stop codons are there: UAA, UAG, and UGA. These three are stop codons. If any one is present, at least if any one is present, then the protein synthesis, the translation is stopped. Okay, so this is what here present. At the 5' end, the start codon is present. Start codon is invariably always AUG. No other codon is called as start codon. Start codon AUG codes for methionine, whereas the stop codon at the 3' end could be UAA, UAG, or UGA. Understand? So this is present in the mRNA. Then now at the center part, this is the main thing in the mRNA. This contains the coding region information from the DNA. Understand? So the coding region contains the codons, triplet codon. It could be anything, A, T, T, G, T, A, anything. Okay. So accordingly, the amino acids will be added. So accordingly, protein synthesis will occur. Understand? So this is the structural genes. This codon region is called as structural genes. So these genes are called as structural genes. They only decide which amino acid should join together and which protein has to be formed. That's why this is called as structural genes. Okay. So this is the structure of a mature mRNA. Now let us see the differences between a prokaryotic mRNA and eukaryotic mRNA. Okay. So prokaryotic mRNA means here we talk about microorganisms, bacterial mRNA. So here, example for prokaryotic mRNA is what we call as bacterial mRNA. That is called as polycistronic mRNA. Bacterial mRNA or prokaryotic mRNA is called polycistronic mRNA. Whereas eukaryotes, human beings and other higher organisms, that mRNA is called as monocistronic mRNA. Okay, let me write here example human beings. So what is the difference between a human mRNA and a bacterial mRNA? Okay, so first let us see a eukaryotic mRNA. So in the eukaryotic mRNA, if you see here, this is the coding region, right? So which codes for the what protein has to be synthesized. So the complete coding region codes for only one protein. So when it is translated, okay, so in the ribosome, it translated, protein synthesis occurs. Now only one type of protein will be produced from this sequence. Understand? That's why this is called as monocistronic. Monocistronic mRNA consists of single cistron. What is cistron? Cistron means a section of the DNA or the RNA molecule that codes for a specific polypeptide in the protein synthesis. So here, this is the cistron. This codes for only one type of protein. So that's why this is called as monocistronic. Whereas if you see in bacteria, which is polycistronic, here this is the coding region. This coding region contains several different sequences. So this pink color codes for a pink color protein. This blue color codes for another blue color. This purple color codes for another purple color. Examples, okay. So like this, different types of proteins are produced at the same time from a single region. Okay. So that is called polycistronic. Polycistronic mRNA consists of more than one cistron. So within this coding region, many cistron are present. Understand? So this is polycistronic. So monocistronic is present in eukaryotes, whereas polycistronic mRNA is present in bacteria and some type of viruses which we call bacteriophages. Okay. Then here it codes for only one type of protein, single type of protein. Here it codes for multiple type of protein. Understand? So in the monocistronic mRNA, the cell size is big. So there will be a separate place called nucleus. Inside the nucleus, the mRNA will be produced and will come and act in the cytoplasm. Whereas in case of bacteria, the cell size is small. It is single cell. So there is no separate nucleus, right? So production and action, everything occurs at the same site in case of polycistronic mRNA. Understood? Right. So this is the difference between monocistronic mRNA and polycistronic mRNA. But basically, you have to remember one thing, whether it is a monocistronic or polycistronic, the mRNA, they are always short-lived. Short-lived means once it finishes its task, that means once it carries the information to the ribosome for protein synthesis, once the protein is produced, that's all. The mRNA gets degenerated. After that, it won't be helpful. Okay, say for example, so this is one protein. Let us say this is one protein, XYZ protein. Okay, so once this XYZ protein is produced, this mRNA gets degenerated. Suppose if the cell needs some 500 number of XYZ protein. Okay, if it needs in 500 numbers, then the same mRNA won't code for all this XYZ protein. Okay, so in order to have another 499 XYZ, another 499 mRNA has to be produced. Understand? So though it is a same protein, each protein has to be coded by a single mRNA only. That's why this is short-lived. Same thing in the polycistronic mRNA. Okay, so this is a ABC protein, this is a ZYX protein, this is a 123 protein coded. So once this is translated, only one time only these three proteins will be produced. So if the cell wants many copies of this protein, the same mRNA will not come and function here. So another mRNA has to be produced, has to be synthesized by the DNA. Then only the same protein will be produced. Understand? So that's why we call this as a short-lived. Got it? So I hope this lecture is clear for you. And in our next lecture, we will see in detail about the structure of ribosomal RNA, rRNA and tRNA. If you like this lesson, like, comment, share and subscribe to our channel, NEET Biology Expert. Thank you.